Perfect. Okay. So this was a beautiful introduction. Actually, it goes straight into why I was actually excited to talk about it, because I am one of those people who is a consumer of this technology, or I would like to be. You know, I am extremely interested in human cognition. I deeply care about consciousness, language, stuff that, you know, we think that humans are a good model for, and it is much more difficult to do this in animals. So when I started seeing these laminar response, these laminar images, you know, from fMRI and other labs, I got really, really excited. And as you know, one of the things that makes us very excited is the fact that we know that layers, the cortex has all these different layers, but also there is streams of communication. So we think there is feed-forward information, there is feedback information, and those actually have different layers distribution. So for instance, feed-forward activity goes into, feed-forward information goes into layer 4, layer 5, whereas feedback activity tends to actually go into extra granular layers. And this is something that still is not really known in humans as much as in many other animals. And this is a venue where laminar fMRI can really open up a door for us, you know, people interested in human cognition, to try and understand what feed-forward and feedback pathways actually do. What is the typical way in which this has been investigated is through laminar recordings. So these are invasive recordings done mostly in animals, whereby, you know, instead of actually having one electrode that has just one tip, you have a linear array that has many electrodes, and then you can actually record activity throughout all of the different layers. And there is beautiful, there's, you know, my favorite, of course, is all of the work from Charlie Schroeder. He was my mentor, so you know, I cannot, you know, and then he was the one that he was one of the pioneers about that. But there's many more labs that these days actually do these kind of studies. But I also want us to remind us why I think laminar fMRI plays a role, and it's important for us to really push it. And one of the things is because the human brain is not just only larger as compared to the animals of mice, the brains of mice or chimpanzees or primates. Actually, primates have developed particularly supragranular layers. And there's now beautiful data from the lab of Matthew Larkham showing that there is cells in area in layers 2, 3 that actually seem not to be present in other animals and that perform very interesting computations like XOR functions. There's also new data coming from the Allen Institute looking at transcriptomics, demonstrating that there is actually lineage of neurons with certain properties that are only present in primates and in particular in humans. So I think this work is going to become even more pertinent as we go, because even though we tend to actually make these assumptions that, you know, we can just take a cat, we can take a mice, we can take a mouse. Well, actually, to a certain degree, yes, but for other things, maybe not. And if we think that supragranular layers are particularly important, as I'm going to show you later on, maybe we need to take the extra effort of trying to characterize and use laminar fMRI. What I've been trying to do in my past research is to combine research from invasive recordings as well as from non-invasive recordings, in particular electrocorticography and magnetoencephalography, as well as invasive recordings, in this case it's laminar electrophysiology in humans. I haven't done laminar fMRI, but I will tell you later on who has done it and why, you know, like I became interested in that. And today what I want to do is to show you 2 examples as to why we think doing laminar electrophysiology, even though in humans is painful and is difficult, is relevant. And I'm going to show you 2 examples. One of those relates to predictive coding. So why a lot of the human neuroscientists are so interested is because predictive coding, which is a theory that is very popular these days, makes very specific predictions as to where information goes and how information passes across the hierarchy. And there's many people trying to investigate this through non-invasive techniques. But we believe that, you know, here is where laminar electrophysiology plus invasive recordings plays a particular role. And secondly, I'm gonna then do a mea culpa in our own field as to also, you know, when we do electrocorticography, for instance, we look at a particular signal, which is called like the high gamma or the broadband gamma. It's a very interesting signal. It's very selective. This is what every single, you know, human person who does electrocorticography uses, yet we don't know what it is. What is also interesting about that signal is that it correlates very nicely with ball response. I'm going to show you in the second part of my talk research that was done in Charlie's lab by my chin, where he looked at what are the generators of that. and the story is going to turn really interesting for all of us. On the one hand, trying to understand what electrocorticography measures, but also what probably is translate to wall responses. Okay, so let's start. So why to care about the layers, and why to care about the layers in humans? And as I said, most people started with this idea of predictive coding that turned cognition around. The idea here is that we are not just perceiving feed-forward input, a la Hubel and Wiesel, but that in contrast, the brain is somehow, let me just, oops, where do I say? Here. The brain is something like a scientist. It always generates prediction that comes from from infranuclear layers to, it emits those predictions or sends those predictions from higher up in the hierarchy into lower areas, into supranuclear layers. And then what actually goes past or is sent throughout the hierarchy is the so-called prediction errors. What that means is that it's the difference between what the system has predicted versus what it has encountered in terms of sensory evidence. And what is relevant about this model is that it's hierarchical. What this means is that this process goes on along the hierarchy. So let's in this case, let's take V1, V2, and LOC. What this requires, as it will become fairly evident, is that we need to actually have access to multiple scales, which is what Camille was actually saying, right? So minimally, we need to have access to circuits, areas, of course, and to layers. And to do that, what we have tried is to combine electrocorticography, which surveys a lot of portions of the cortex. And this is a particular, it's one of the things that NYU is, where these recordings took place, that's different from many other centers. That this is electrocorticography. So this is a large survey of many, at least one hemisphere, plus combined with, these are what I showed you before, these are these linear arrays that we get ethical approval from the RB as well as from our subjects to actually do these studies. And so what do we investigate in this? For this particular study, we tried to look at 3 components of the so-called predictive coding model. On the one hand, the idea from predictive coding is that if there is a very strong model and a very good model, then the prediction error is going to go down, right? So in essence, it can predict very well what the sensory evidence is giving me. Therefore, the so-called posterior is actually very low. Now, if the model is bad, I'm going to have a very high prediction error, and then this is going to trigger the so-called model update, right? And then as the model gets better, then the same process goes on again. So the prediction errors are reduced if the model was good, and so on. And so how did we investigate that? It was a simple mismatch negativity model. So in this case, we present 4 different syllables, something like pa, 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 ga. And the last one can either be something that the subjects can predict, because it's the same syllable as before, or it actually is something different. So pa, 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 ga would be the deviant, pa, 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 pa would be the same response. And these are when you take, I think these are 11 subjects that we have. As you can see, this is not fMRI, but if you combine all of the subjects that we have, actually we can survey the brain in a fairly good manner. And what I will show you before is which ones are the areas that show prediction errors. What does that mean? Areas that reduce their activity as a function of showing the same stimulus. And the hypothesis here is that these are the areas that, because they generate a good model, the response is reduced. And what I'm showing again here is the so-called high gamma responses, which, as I told you before, they were thought to reflect multi-firing rate, but the story is going to be more complicated and more interesting because they might actually reflect dendritic integration. So what you observe here is that, first of all, these areas in superior temporal gyrus and posterior superior temporal gyrus tend to have stronger reductions in the responses. This is what you can observe here. One electrode, the one that's shown here. The gray line are the single trial responses. The red line is the average response. And as you can observe, the amplitude goes down significantly as a function of presenting the same stimulus. What is very interesting is that despite the fact that the responses go down in amplitude, the classification is not affected. So in this case, the adapted responses are actually equally, if not better, classified than the non-adapted response. And this is something that the group Peter Koch has actually shown before with fMRI. All right, so now the question becomes, so what are the areas that respond to changing of these priors. So if I show the pa 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 ga, right? And so here what you observe is that these areas that I showed you before that would reduce the responses significantly now respond very prominently. This is what we interpret as areas that show prediction errors. Now there is this other set of areas that are actually very interesting. Why? Because they do not show any response for the first stimulus, for the second, or for the third, but only response at the level of the deviant. And these ones we interpret as actually being prior updates. The idea here being that the subject has to change to a new model when the prior is presented. Excuse me, when the deviant is presented. Now you may ask yourself, so how do these two areas interact? Do they have any way of interaction? They do, so we try to investigate that through the so-called Granger causality, in this case on the high gamma. So we took these two different responses, what we call like prediction errors and what we call prior updates. And the idea here was that, or at least it was our hypothesis, that when there is a good model, then there is the information flow would go from the prior, so to speak, or from the model to the prediction units, whereas in the case when the model is bad, the information would flow the other way around. And that's exactly what the Granger causality actually tells us. This is what is shown here. This is what is shown there. Now, whether or not this is really true, this is what the Granger causality says. We need to probably think, more and conduct more analysis trying to characterize these two different areas. Okay, now the question became like, are these real so-called prediction errors? And one question that you may ask is, could they just not reflect, for instance, adaptation, like simple and trivial adaptation in, like neurons? We don't think that that's the case because we conducted another experiment where in this case now we switch to tones. And now what you can do is a situation whereby you can either repeat the two tone and it's predicted, or you can repeat the same tone, but it's unpredicted. So in this case, the level of adaptation is essentially the same. The only difference is one is predicted and the one is not. And what you observe here is that now the unpredicted tones elicit much higher responses than the predicted ones. So we think that the computation is actually active. It's not just a passive computation. All right. But what we really wanted to look was what happens in the layers. So predictive coding model has this hypothesis that the prediction errors should be mostly computed in supragranular layers. Or put it differently, that's not true. There's certain models of predictive coding that postulate that prediction errors should be computed in supragranular layers. Some models actually do not assume that. And what I'm showing you here is, so this is a craniotomy. These are two electrodes that are from the large ECOG grid, and this is the tiny laminar electrode that we implanted. This is, you can see it here. So it gets very thin, a really, really thin nail, so to speak, that has like 24 contacts. And this is just to show you, so one of the problems, so to speak, with laminar electrophysiology in humans is that it's opportunistic, right? So you cannot go and insert the electrode where you want. It has to be in an area that's likely to be resected. And hopefully you hope that it's going to have functional responses as you expect. We were lucky enough, and this was exactly the case. So if this is the electrode 21, that as you see has no response to the stimuli that we presented, this is the electrode 29 that has a strong response to our stimuli, and luckily the laminar electrode was actually close to this electrode, right? But again, this is, you have to be lucky as well. So it's not; these are not things that these are not monkey physiology where you can target a particular area. All right, so you already have seen CST profiles. So this is what I'm showing you here is cortical depth. This cortical depth, this is time when we present the first stimulus, the second stimulus, the third, and the fourth, either in a condition in which the four of them were the same, or when the last one was a different one, right? And these are the so-called like current source density profiles. You can, they reflect sources and sink in terms of current sources. What I want you to take from this is, first of all, the initial responses that, very much represent this. You can also see here the strong superficial response that actually goes down as a function of repetition, as well as a recovery of the suppression from the deviant one, right? And this is just one single patient We implanted these laminar electrodes in 5 of the patients at NYU. And what you have now, what you see now here, this is a reliability map. So this is in how many cases from the patients that we have investigated, we observe responses in this particular cortical depth. And what you can observe here, this is, like around 4 to 5 patients have a response in this area that is marked here, right? So the responses are mostly supragranular. Again, caveats, as I mentioned before, these are, even though these are, probably as good as it gets, we don't have the capacity to really align them across the subjects as much as we would want in a penetration, for instance, in animals. But at least we can actually characterize them functionally. So that's certainly one advantage. All right. So I hope that with this, I at least want to show you that it is possible to investigate laminar responses in humans. It's hard. It has its caveats, but it can be done. But then I want to tell you another story, which I think is also very important and interesting, which is, as I mentioned before, most of the community who does invasive recordings in humans, they tend to, focus on the so-called like high gamma activity or broadband activity. Why? Because Ray and colleagues already demonstrated that there's a strong correlation between multi-unit activity, as you can see here, and the so-called like high gamma, right? And the correlation tends to break if you look into in this case either all frequency bands, for instance, such as in this case alpha. So for the most part, the community has just assumed that what we measure is just multi-unit firing, and this is what you will read in any paper that people from invasive electrophysiologists published. It's also interesting because when people have done experiments whereby they have tried to correlate these different frequency bands, for instance, in ECOG and fMRI, this is one particular paper that I'm just citing, but there's plenty of those, demonstrate that the correlation with the high gamma and the fMRI is actually much higher across time than any other frequency band. Right, so most of us are really excited because we think that the high gamma is a good marker, so to speak, or it's a good translational signal to the fMRI response. Now, and this is work that Marcin has done, in Charlie's lab. So I'm just, like you need to credit him, for what I'm showing you here. So what Marcin did was he took recordings that that were already taken in Charlie's lab from either V1 or from A1 in monkeys. I'm going to show you first results in monkeys, and then we're going to go to humans. And these are, again, CST profiles. Can you see? CST profiles, they use diffuse light. Why? Because it's known that they activate the supragranular layers very, very little in terms of spiking. And now the question became, okay, so if multi-units are really the same as high gamma, then you would expect minimally the same spatial and temporal profile, correct? And that's like simple. And what you observe here is in color in the background is the responses in the high gamma, and this trace here represents the spiking. And you can just see naked eye that it seems that in infragranular and in granular responses, the high gamma tend to correlate, whereas in supragranular, actually it is not, the spiking is not present. The same thing actually happens if you go to A1 and you show broadband noise. Again, in granular layers and infragranular, there's a strong, there is some correspondence between the spiking and the high gamma, but in supragranular layers, there is none. And if you quantify, that statistically, so in this case, it's the statistical map as to where in the different layers and in time the MUA actually happens or the high gamma, you can see that the correspondence between these two is actually first of all, there's two things to note. One is that they are mostly in granular and infragranular layers. And second, the time at which they tend to correlate is actually much shorter if you compare with the supragranular layers. Furthermore, if you go into auditory domain, you can see here that there is an early response, again, but very short, in which they tend to overlap, but there's a much more sustained response in which they don't tend to overlap, right? And this is just another quantification, just for you to visual to make it very clear. Now they are, this is high gamma, this is a broadband response for different layers, so either for granular, infragranular, or supragranular. And this is the same, but for the MUA. And you can see again, naked eye, that the response in terms of spiking tend to be much higher in the infragranular and the granular layers. But in contrast, for the high gamma, it's much stronger in the supragranular, right? And this you could say, well, but this is just probably just diffuse stimulation, right? Well, that's not the case, because they took another recordings from another paradigm whereby they had this is free viewing, so the animals are just like glossing over different stimuli, and again the same dissociation occurs. So much stronger responses in the high gamma for the supragranular, whereas for the MUA the responses are much stronger in the infragranular and in the granular layers. Now, what about auditory cortex, right? Then you could say, well, maybe like vision is V1 is special. This was in V1. What happened with A1? So A1 is actually particularly interesting. Why? Because as you could see, as you could glimpse from the other plots that they show you, the initial response for the high gamma tend to actually be very similar for all the different layers. But lately, the response is actually stronger for the high gamma, and it's exclusively in the superficial layers. Again, the same dissociation, however, with much stronger responses in terms of spiking in the infragranular and the granular layers for auditory cortex. And this is not only when you show broadband noise, but even when you go to best frequencies. So when the cells are actually the strongest activated, it happens exactly the same, right? There's this later response in the supragranular that reflects most of the high gamma, and there's again this association with the spiking. So now you could say, well, but these were monkeys, right? So how about humans? Well, so here you can see actually human responses. These are human responses from human prefrontal cortex. This is contrary to the monkeys. These are just resting state responses. So whatever just happens at rest. For broadband activity, so this is the high gamma. For supragranular, infragranular, supragranular, infragranular, supragranular, granular, and infragranular. For broadband activity as well as for MUA, right? And what you can see again is the same responses I showed you in the monkeys. So much stronger activity for the broadband responses in the supragranular, whereas in contrast, the MUA is the strongest in granular layers, which is what you would expect. Is this in one particular subject, in one particular electrode? No. You observe the same response, it's systematic, at least across two subjects, right? Still, of course, you would want to have more data, but at least these two subjects actually point to that, to that effect. So what would I call as promises? I think that there's something really cool about this new data. Not only the fact that that we can measure human responses in human, that we can measure layer responses in humans with electrophysiology, that we have, that we can show that there is prediction errors in supragranular layers, but also I think I'm particularly excited about these broadband responses and probably the relationship with fMRI, because if it's true that they reflect and predict this, we are onto something really cool, and it would be something very interesting because it will be particularly targeted to feedback, right? And as I showed you, and as I told you before, one thing that we know as well is that in humans, layer 2-3 is particularly large as compared to other species. Oh, okay, but what are the challenges? And I think that I just want to remind us, as to what we all know, but. So this is data coming from Matthew Self. He ran a figure ground segregation task. In short, what he discovers, which is actually very concordant with what one would expect based on the connectivity, that the feedback responses are mostly in layer 2-3, as well as in layer 5, which is exactly where the feedback responses actually should come. However, from the human literature, Camille already presented part of this data. The picture is not as clear, right? So Lars has shown that predictive responses are mostly in supragranular layers. Peter has shown that they are mostly in infragranular layers. I think that this is something that we just need to reconcile. There's something that needs to be done here. So at desiderata, I think that it would be great if we could actually have invasive methods such as laminar electrophysiology in humans, and at the same time do that, with laminar fMRI. And I really believe that we should keep these kind of get this kind of maps in mind, because you might, a lot of I know that CMRR has produces technical advances, but I think that we can use those technical advances to then understand the human mind in ways that otherwise are completely not permitted. So I think we need to really keep in mind that there might be something special about supramolar layers in humans and in primates as such. So that's just something to kind of keep us trying, because we might be onto something. So, and maybe that desiderata is possible. So Esa and Oren and Federico and Camille and Elia and myself, we got a grant from the Brain Initiative to try and do this, but COVID came along. So we hope that, one day when COVID is just not there, we will be able to really, put this together. And with that, I just want to thank you and actually the people that really did the work. So Machin, Charlie, and Eric, with whom actually we did the Laminar recordings together, and then Kasper, Wolf, Istvan, who actually made these electrodes in humans. And this is the NYU team, the neurosurgeon, Oren, and Thomas, and also Richard, who's been instrumental in this. So I'm sorry if I am past. Okay, that's it from me.